Ned, thank you so much. That was a magical talk, and what a talk to end on, really. And with those pinhole images of the eclipse, my goodness, that truly is magic. I said about wonder at the start of the day, the wonder in a bee orchid, and we've ended with an absolute show of botanical wonders. Thank you so much. And the comments that have been coming in through the chat are just amazing. You really have brought imagery of plants to an art form this evening. So thank you. And it was great and somewhat ironic that we began with Goethe in the form of Goethe's Camerops humilis, that great ancient palm in the garden at Padua, and you've ended the day referencing Goethe, and what a great sort of round circle of the day it's been. There are plenty of questions. A lot of the comments were coming through about those amazing bark and cones, which of course has been a highlight of your blog postings from Harvard, especially during the pandemic and the lockdowns, which have been great inspiration to me, and I'm sure many thousands of people that have looked at them. Those incredible colors of cones. And when you showed the Himalayan fir slide, I just gasped, as you probably did when you saw it. Quite extraordinary. Well, there are some questions, and they're some quite obvious questions. Starting off with the bark, Ned. What are does anyone know the functions of those different colored bits of bark on the same tree, and maybe the variation in bark color that we see?
I don't think we do. I think the greens, all smooth bark species of trees always have a photosynthetic layer of bark under the outer dead bark. So I can say that as bark gets pushed out into the sort of outermost layers, you could imagine it coming and emerging with greens at times. But at other times, the oranges, these what I assume are potentially carotenoid pigments, I don't believe people have studied them in any way functionally. So I think it's just fascinating. I think that's part of what makes me enjoy it so much, is I have no idea why. But as with so much of biology, there are a million ways to do things, and for some reason, all of them or many of them seem to work. But the answer is I certainly don't know. And I will just tell you, it's anecdotal. I just feel that bark is most intense in the winter, and it may just be because the sun is more intense and you're getting a little bit lower angle and the leaves are off the trees, but they really do seem to glow in the winter. So unfortunately, I don't know.
Well, that's good. It's good to not know the answers, isn't it? That's why we're biologists, to find out. And then perhaps something you can answer then, the vicin threads. We've had a couple of questions in. What are the function of those vicin threads in the rhododendrons?
They haven't specifically been studied, but the assumption is that this is one of these things where aggregate groups, larger groups of pollen grains going on one pollinator and moving for pollinator efficiency to another flower is the case. Certainly in azaleas, there have been some studies that show that moths and butterflies move these strings of pollen grains quite effectively. I think it would be a wonderful thing to do some real pollinator studies. This would require, in the case of this species, probably working in China. One thing we did do, I didn't show the video of it, we brought these in and we did some very high-speed videography, several thousand frames per second. And we then took a vibrating mechanism and tuned it to essentially a bee that does buzz pollination, about 220 vibrations per second. And we just had fun. I don't know that bees pollinate this the species or not, but I can tell you that when you buzz these things at those frequencies, you see this magnificent set of shards of strings of these little strings, and they're just flying out, and they will almost certainly hit the insect right there. We did some videography in the living collections following bees that did come to our rhododendrons, and you could see buzz pollination, and you could see these fragments, really just hitting the bee everywhere. So it's certainly an evolved pollination strategy. And I do know that most of the rhododendrons that I've tapped, it's rare that you'll get something where the entire content of the anther comes out at one time. Mostly you'll get bits and pieces of shorter pieces. But if you have Fortunii, and I don't know that you do in Oxford, please visit it. It never disappoints. And it's got the weirdest bubblegum smell that you've ever, sort of encountered in a rhododendron.
Yeah, I think we do have it at the Harcourt Arboretum. So
okay.
I will go and look at these rhododendrons in a completely different way now, Ned. And just following on from that, when or do the tetrads separate into individual grains, or are they also?
I think they do. They'll be dispersed as tetrads in rhododendron, and then they will arrive on the stigmatic surface as tetrads.
Okay, I don't think we have. It just keep that there's lots more comments coming in about how beautiful everything is, rather than specific questions.
That's fine. That's all I intended, was to remind people that the botanical gardens of the world are the place where the world should meet plants, as well as, of course, the natural world. But for so many people who live in cities and who don't have access to nature, gardens are where organisms are front and center. Certainly.
And what a great note to end on. So, Ned, thank you very much for that pièce de résistance at the end of what's been a long but amazing day. And thank you to all our speakers today. And we look forward to seeing everybody, or as many as possible, again tomorrow as we continue this symposium. So thank you all, and thank you, Ned, and we'll see you tomorrow. Bye for now.